There it is. Okay. Hello, everybody. Hi. Welcome to this talk from our 9th annual Women of Science event. This morning, we are very lucky to have 2 speakers with us who are going to be dividing the time together. They both happen to be from the same research institute, which made pairing them together an excellent and natural fit. But we will be starting off with Hilary, and then we'll be hearing from Swati afterwards. So I will first introduce to you Hilary Johnson, who is a Master of Science student at Université de Montréal in their Institute for Research in Immunology and Cancer, which is IRIC. And she is studying the genetics of leukemia and hopefully having a great time with it. She's going to tell you all about it. She was born near Toronto. She completed a BSc at Western in London, Ontario, and at that point discovered love for the creative, diverse, and exciting field of science and biology in particular. And she's passionate about women in science, which makes it great for her to be speaking here today. And she also enjoys cycling, music, art, lots of stuff that she's going to be speaking to you about. So I am going to be monitoring the chat, and I am going to be monitoring the participants as well. So if you have any questions, feel free to enter them in the chat, or you can raise your hand, and I will be moderating both of these and interrupting as necessary to identify to Hilary that there are questions. So feel free to ask at any point. There's going to be a first part, and then there's going to be a pause for questions. But after that, feel free to interrupt at any point, and I'll come in and let Hilary know that you have a question. So without further ado, Hilary, take it away.
Okay, thank you so much, Carl. So I'll share my screen. Okay. And you can hear me okay and everything?
Yes.
Okay, great.
Okay.
Okay, perfect. Okay, hello everyone, nice to meet you. I'm very excited and honored to be here today. As Carl mentioned, my plan is so for about 10 minutes or so, I'll speak a little bit about science and my research, and then I'll probably take a pause for questions if there are any. And I think it makes sense to kind of pause midway through, because if questions arise, they might become more clear as I continue the story with science. But then for the second half, for 10 minutes or so, I think it'd be nice to speak a little bit about my experience as a woman in science. And I think from that point onward, it could become a discussion if you guys like. And I think it would be fine if you could interrupt me with any questions that you have. But I think I'll probably let Carl stop me or anyone if they have questions. Otherwise, I might not hear or see any that pop up. But regardless, as mentioned, my name is Hilary. As Carl mentioned, I was born in Toronto, and wanted to speak a bit about this because, I was born, outside of Toronto, and went to high school there. And I think this was an important time for me because when I was in high school, I didn't have a ton of courses to select from. I kind of had business courses, science courses, and the arts, but I found particularly, I really enjoyed my science classes, and something that I'll talk about a bit later, but I think I really clicked in them because I had female teachers. So I had a female biology, chemistry, and physics teacher, and I think this was a really great atmosphere to be comfortable and approach them and develop nice relationships with them, and kind of encouraged me to pursue a career in science. But specifically, this led me to do, as Carl mentioned, a Bachelor of Science, where I specialized in biology at Western University. And I think it was nice to choose a general specialty if this might help anyone with selections academic-wise in the future. But I could kind of, what was nice about Western was remain kind of general. And so within biology, I could choose a bunch of different subtopics. So I took courses in genetics and cell biology and physiology, evolution, all these cool courses. So that kind of allowed me to choose what niche was best for me and what I really enjoyed the most. And so after the 4 years of my bachelor's, I started thinking about what was next for me. And that was when I got an email from a guidance counselor about an internship opportunity at this institute that I work at now, the IRIC. And I applied for this internship. And then the summer of 2019, I did an internship in a lab where we studied histone proteins, which are these proteins that help package DNA into smaller structural subunits within the cell. And so in this context, we worked with yeast, and this was a great kind of place for me to start, I think, because they're very easy to work with. But I kind of knew at the time that this wasn't exactly what I was most passionate about. So I started talking to people at the Institute, and this is a couple pictures of where I work. It's a really beautiful research building. But I was talking to my one friend, Kyle, and he sounded really exciting about his research. And he said, hey, like my professor is really interesting, and he's has a lot of success with clinical trials, and he actually has this molecule which he's using in clinical trials, and it's shown a lot of promise in treating patients with leukemia, which is a blood cancer. So I asked him a little bit more about it, and turns out he was working in the Sauvageau laboratory where I'm working now. And the molecule he was talking about is called UM171, which is short for the University of Montreal, where we work. And I think it was 171 because it was the 171st drug that they screened in a chemical screen across leukemic samples, but or across blood samples. But essentially what they found, and I'll go into detail to explain this a little bit later, but they found that this molecule could be used to expand this specific type of cell population called stem cells to be used in the treatment of blood cancers such as leukemia and other blood disorders, now recently discovered that it could be used to treat in. So I knew a bit about stem cells from my bachelor's, but for those who don't know, I like to think of stem cells as kind of a blank slate or a blank canvas cell in our body, in terms of the fact that there are very few numbers of them in our body, but they're super important because they don't have an identity. But when they're given cues or signals from the body, they can turn into kind of almost any cell in the body to do their job, so that, or they can turn into any specialized cell with a specialized function. So for example, if a cell with a function that like has a role, and this cell, let's say, dies or becomes mutational and is no longer functioning, then this stem cell can kind of come in and replenish the malfunctioning cell and take its place. And so they're super important in the body, and it's kind of a one-way street. So this stem cell can become a cell with a specialized function, but if this cell that's identified and has an identity, if something goes wrong, it can't go back to a stem cell state. So that's why these cells are very important. But regardless, once they turn into these cells with a function, they can circulate the body to do their job. And what's interesting is that there are many types of stem cells. So there are epithelial stem cells, which turn into your skin, which line your blood vessels and urinary tract, as well as neuronal stem cells, which turn into the cells of the brain, mesenchymal stem cells, which turn into the cells that make up your muscle, bone, tissue, and fat. And then finally, hematopoietic stem cells, or HSCs, which reside in the bone marrow and your blood, and then turn into the cells of the blood and immune system. So I have hematopoietic stem cells bolded here because this is the type of cell population that this UM171 molecule works on specifically in the treatment of leukemia. So I'll also speak a bit more about what leukemia actually is, because I learned quite a bit of it when I started working in this lab, and I think it's really important for the context of my research. But so leukemia is a blood disease or blood cancer, and the way that it starts is that, to understand the body, and I'm not sure how much knowledge you guys know, but I think it's always good to start from the basics. But essentially, the cells in your bone marrow are where these hematopoietic stem cells reside, and they are responsible for turning into your blood cells and your immune system cells once they leave the bone marrow and start circulating in your body. However, the mutations within the hematopoietic stem cells and the cells within your bone marrow that cause these cells to be no longer functioning means that healthy blood cells and healthy immune cells can no longer be produced. And then you start to have an accumulation of these malfunctioning cells within your body, and you're no longer able to produce immune cells. So you're no longer able to fight off the common cold. You can no longer produce blood cells. So your body can't transport oxygen properly. And this is why this is such a fatal disease, unfortunately. So the way that in which leukemia is treated is, first, patients are subjected to a chemotherapy, so a drug which destroys the cancerous immune and blood cells within the body. And then, since this patient is deprived of an immune system now, what we do is in the lab, we'll take a healthy blood unit from someone who was healthy, and we can in the lab treat it with this UM171 molecule to expand these stem cells, which are super regenerative and can be used to help the patient, so that when this blood is then transplanted back to the patient, they have all these rich cells, all these like blank slate cells that can come into the body and then transform into a healthy blood and immune system and replace the lacking blood and immune system of the patient with leukemia. So the way in which this molecule is so incredible is that when we add it to blood in the lab, we can then create this super healthy and rich blood sample, which can be used to treat leukemia and other blood cancers, as well as anemia. And even recently it was found to treat a sickle cell disease, which is an immune disease, and in which the lungs kind of flood with a mucus and fluid. And so because of the success and the use of this molecule in clinical trials, the director of my lab, Guy Sauvageau, then created this company called Excelthera. So this was a really interesting point for me, because the discovery of this lab was exciting in terms of the fact that it not only touched on my passion for stem cells and their regenerative potential in medicine, but also it was this biotechnology company, and I've always been really interested in biotechnology. It's a kind of a new industry that's really blossoming lately, and there's so many different companies coming up all the time, and I think there's so much to learn and so many opportunities in this field. So this was definitely something that I was interested in. So I talked to him and we had an interview, and thankfully he welcomed me into his lab. So I started in his lab in the winter of 2020. It's a picture of our lab a couple of years ago in the top right. And so to give you a bit more idea about what we do, so we specifically work to look at the surface markers of hematopoietic stem cells so that we can target them and expand them for clinical purposes. And we also look at discovering new therapies for acute myeloid leukemia and blood cancers. So we do that by screening drugs against these samples, as well as looking at their genetic profile. And then we'll look at their DNA and RNA expression through single cell sequencing and transcriptomic analysis. And so my project specifically is kind of a blend between this research of leukemia and the healthy blood system. So I specifically look at this gene called the hepatic leukemia factor gene, which has found to be very specifically and highly expressed in hematopoietic stem cells and can be used to identify these cells. However, despite its relevance in healthy hematopoietic stem cells, it has also been observed in leukemia. So my question was to address what are the genes that regulate the expression of this Hlf gene in leukemia specifically, and what causes a transition between a healthy blood system and that in a cancerous blood system. And so since blood is difficult to work with in the lab, I've been working with these cancer cells. And the way that I do this is I can introduce this green fluorescent protein. It's just called kind of ZS green to be fancy in research. But using this technique called CRISPR, I can introduce this fluorescent protein beside the Hlf gene so that when Hlf is expressed, this green protein is expressed as well. And then I can track its expression visually. So I introduced it using this new technique called CRISPR, which actually won the Nobel Prize in Chemistry in 2020 and was awarded to 2 women, which was really exciting. And that was the first time that this was done. But essentially, it's a really fascinating technique in which we can use the specific protein to actually correct and edit and change the genes within cells and even add genes such as this green protein. And so it has tons and tons of application in medicine, specifically in diseases such as sickle cell anemia, which is just caused by a simple mutation. So we're beginning to correct these mutations with techniques such as CRISPR, which is really fascinating. But regardless, on top of using this technique to introduce a fluorescent green protein, I could also use it to kind of knock out or break certain genes. So that what I wanted to do is within these cells, which are very disposable and harmless in the lab, I could break one of a genes that I think could regulate this Hlf gene. And then if I knocked out this one gene and I saw a loss of the green signal, then that would tell me that this one gene regulates my gene of interest, and was a really great starting point to kind of determine how this gene network is in place and how it functions in leukemia, which I ultimately have the goal of comparing it to a normal blood system. And again, determining the differences between the genetic network in leukemia to that in a healthy blood system to determine what actually happens that leads to leukemia and how can we prevent this. But essentially, regardless of understanding this genetic nature, I also know and I think that this CRISPR strategy can be used to track the expression of any gene of interest. So I think it's really something that's exciting and promising in regenerative medicine. But regardless, I couldn't be here within my master's so far without mentorship from my supervisor, Bernard Leonard's on the left. He looks kind of scary, but he's very nice actually. And then the research director, Guy Sauvageau. But this is kind of what I have to say about the science that I've done so far. And so if there's any questions at this point, I'd be happy to take some, but if not, I can kind of move on into the importance of their mentorship and such in my project.
I'm not seeing anything at this point.
I raised a hand.
Oh, you did? Oh, there it is. What do you know? Sorry.
Background is orange too, so is the hand.
Hi, Hilary. You had mentioned, I think, unless I heard wrong, that you said CRISPR is being used to treat sickle cell anemia?
Well, no, it's not actually, sorry, excuse my language. It's not to the point where it's, it can be delivered to a person who has sickle cell anemia, but there is quite potential shown for cells in the in culture in the lab and in mice models. So you're right, it is not at the stage where it can be used directly in humans, but there is quite a potential for it to get to that stage. And that is, of course, the goal is to eventually bring it to a stage where we could use it in humans. But no, excuse my language at that point.
Do you know any of the studies that are being done? What do they study the gene in vitro? Do they just edit it and they put it into some kind of human culture of cells? Do they study it in another organism?
Well, I imagine, I don't know too much about sickle cell disease, but I know that, for example, in leukemia, you could take, what we do is we go within, excuse me if anyone's squeamish, but we kind of go in with a needle into a patient with leukemia into their bone marrow and then take the cells from there and culture them in the lab and then apply these techniques in the lab. And so I imagine they would be doing that with a patient with sickle cell. So they could maybe take a sample from their lungs and such and then investigate that this mutation in the lab in culture, so ex vivo. But also I would imagine that similar to leukemia, you could induce a sickle cell state in mice and then kind of try to correct this mutation in mice and try to see what the effects are at that level. But I know it hasn't reached the human stage yet because there's quite a bit of complications in that.
Have you been using mice as a model organism in your experiments?
I haven't at that point because I have lots to do with cells, but we have done, I mean, my supervisor's done a couple of transplantation experiments, kind of determining what the effect is of transplanting cells with only HLF expressing cells and then no HLF expressing cells. And they're a bit inconclusive. We'd like to repeat that, but it is, I mean, hopefully something that I'd like to do soon.
And I'll ask one more question because I guess I'm monopolizing it. What is normally the function of HLF when it's not?
No, it's a great question. In the blood system, it's been shown to kind of, so basically these stem cells, they can either be in a quiescent state where they're not cycling and they're just staying dormant and quiet until they're needed, or they have a state where they're turning into a cell that's needed. And so this HLF gene has been shown to kind of keep them in that quiet state and just kind of sleeping, if you would, before they're needed. And then in the context of leukemia, it's been shown to kind of induce a chemo-resistant state in cells. So when it's highly expressed, they've seen that cancer cells are more resistant to treatment with chemotherapeutic drugs. So and it's also been shown to kind of prevent them from going into apoptosis, so prevent the cells from dying. And so that's definitely a relevant case for how its function works in leukemia specifically.
Okay, cool. So they keep the cells from dividing. It's like a lot of the oncogenes that we study, I guess. It's just that checkpoint in cell division where we're like, this needs to be working for it to stop. And presumably a mutation in this gene is what cuts the brakes and
sure, that could happen. Yeah. Or a mutation in something that regulates it, like for example. So yeah, so there's a lot to learn about it, essentially.
So cool. Thanks.
Of course. Thank you.
I wanted to point out quickly, now maybe some stuff has changed even more recently, but I remember hearing something about this and I just went back and double checked, and clinical trials have actually started with sickle cell anemia, or it was announced that they were about to start with sickle cell anemia earlier this year. So it's possible that this could actually be taking place with humans as of right now. But there is a question. Hilary, could you just take 2 seconds and describe really quickly what CRISPR actually is?
Yes, of course. I know I kind of went through that quickly because I wasn't sure how much time I would have, but I would love to. So basically, CRISPR is kind of two components, and an analogy that I've read that I think is really interesting and really accurate is that imagine you have a train with a conductor at the beginning of the train. So the CRISPR has two elements to it. And so one of them is the train and the other part is a conductor. So the conductor is what you would consider this protein called the Cas9 protein. And so this protein actually, I would consider the train as the Cas9 protein and the conductor as this thing called an sgRNA. So the sgRNA will guide a protein to a specific site in the gene, and then the Cas9 protein or the train will do its job, which will cut the DNA. So essentially the way it works is that, let's say you have a strip of DNA with a mutation, then you will design something called an sgRNA, which stands for a single guide RNA, which matches that DNA, so that when you introduce this sgRNA into the cells, it will find its way to the DNA and match up with the DNA. And then you have this Cas9 protein, which is a second component, and the Cas9 protein will come in and it will bind to this sgRNA, and then it'll induce a double-stranded break. And then what happens is that, so a double-stranded break is made, and then through the mechanisms of the cell, this break will be repaired. And so either, like I've mentioned before, there are different purposes of CRISPR. So you could use it to induce a mutation and destroy the function of a gene, or you can use it to replace a gene with a functional one. So if this sgRNA is designed so that it has the correct sequence of the gene, and you design it to bind towards a malfunctioning one, after CRISPR creates a double-stranded break, then the mutational gene can be swapped out with the sgRNA through homologous repair during the DNA repair process. So it is a bit complicated, and it's hard to describe without illustrations actually too, essentially, but you kind of have this this enzyme, which is a Cas9, which does its job, and it's like the meat of it. And then you have this this guide or the conductor kind of, which is this DNA sequence, which gives it kind of a template repair to either, to replace a malfunctioning gene with a correct one, or like in my case, to introduce this ZS green protein. So even if you look at this bottom left panel, you see like the HLF targeted locus. So you have the stop codon of HLF, and then the repair template, which is the single guide RNA, includes this IRS element for proper integration, and then the coding sequence for this green fluorescent protein, and then some other kind of fancy stuff for proper integration within the cell. But I hope that that's a little bit of makes a little bit of sense. But of course, there's tons to look up about it as well if you're interested to read more about it. So.
Okay, that's great. I don't see any other questions. Let's keep going. Oh, sorry, hold on. There actually one just popped up.
No worries.
Can you fix a malfunctioning gene, but at the same time mutate a correct gene?
I think that there's a little bit of work done in this. I don't know about doing two opposite things at once, but I know that you can complex this Cas9 protein with multiple single guide RNAs. That's definitely like a new technique that's coming up. So for example, if you wanted to knock out 5 genes at once, then you can complex 5 single guide RNAs to this one Cas9 protein and introduce it into cells. And that's kind of an efficient way to get a lot done at once. But I'm not sure if you could, do opposing things at the same time. But
I think the question here, my interpretation of Jonathan's question was more along the lines of, can you do, as you're trying to fix something, can you do damage as well?
Oh, like unintentionally, you mean? Okay,
Mm-hmm.
I see. Yes, that's definitely an issue right now. And that's why this hasn't moved to the stage with work in humans yet, because there's something called off-target effects, where this Cas9 protein could bind somewhere where it's not supposed to. And, it create a mutation, create a double-stranded break where it wasn't designed to. And so that's something that we need to take into consideration. And that's why when designing the single guide RNA, we need to be very specific, and it needs to be long enough, and it needs to have the right parameters to bind where it needs to. But no, it's a great question. So that definitely happens quite frequently. I want to say in 30% of cases that could happen. And so that's why a lot needs to be done before it can be used in humans, essentially. And there's actually a really interesting story, if you guys want to look it up after, where there was this doctor in China who took the embryo of twin babies before they were born. Maybe you had heard about it. And he used CRISPR to mutate a gene within their genome so that they would be resistant to HIV because their father had HIV. And so there was tons of ethical outcry about this, because that's of course not okay for these unborn babies to have their genome messed with by a doctor, but essentially he did have a little bit of off-targeted effects, and we don't know what the consequences of that are, but that's a perfect example of why this can't be done in humans yet. But it's pretty crazy. But it's been in the news for the past couple of years.
What's that, sorry?
No, the story that I just told has been in the news for the past couple of years. So if anyone would be interested in looking that up, I think it's interesting to read about.
Well, Chris, who asked a question just before, mentioned that he's talked about it in his class.
Oh,
actually,
interesting.
and that apparently, yeah, there are some off-target mutations that have occurred.
Right, exactly.
I believe he's in jail now, right? I think they did jail him because he's a mad scientist. He's the very definition of a mad scientist.
I think so as well. I definitely lost his license or in his lab at least, I think so.
Okay, would you like to keep going?
Yeah, I would love to. I mean, if anyone has questions later on, they can, I think I put my email at the end if anyone wants to email me or stop me later, we can chat later. So that'd be great. So like I said, I've had some great mentorship that led me to continue in my master's, but I wanted to speak about a couple events which I thought everyone should know about in the context of this this recognition of women in science. And so one thing that I wanted to address earlier is that, so I had this mentorship from these two men, and I remember working with them about going to this conference that I went to at the United Nations a couple of years ago, and hearing from some speakers and them talking about not only is it important to encourage women in science to have women mentors, but also men mentors, who are just as equally as important. And so that kind of carried on with me. But regardless, I think this was a conference that everyone should kind of know about, and it happens February 11th. It's a free registration. And so I just registered and then flew to New York City and went to this event and heard from scientists, doctors, as I wrote here, astronauts and other leaders in the field. And it was a really exciting opportunity. And so I think this year it's going to be online, but it could be worth checking out to register if it's not too late. But I learned lots about it. I'm sure you guys have heard these statistics before, but the fact that 30% of researchers in science are women, despite making up, I think, even more than 50% of the students in master's and PhD graduate programs. There is still a little bit of a pay discrepancy, I believe, despite a majority of healthcare workers being women. But at this conference, I think it was really interesting because they talked a lot about mentorship and the importance of having male mentors as well, or a mentor who, like, regardless of gender, to kind of welcome women into the field and tell them that they are important to be there. Like I mentioned before, it was really important for me to have female teachers at a young age to kind of feel welcome into exploring this field. I think it's important to have, many relationships of people who you can confide in. So I have my two thesis supervisors, but there are also many people in my lab who I can approach for advice. You want someone who can communicate with you that they're available and wants to make time for you. And I heard this nice story about someone who had a mentor, and at the end of their mentorship, before that person went to find a new career, they said, thank you for being my mentor, to their mentor. And their mentor said, well, thank you for being mine. And I thought that was really special, because you want someone who can learn from you just as much as you learn from them, essentially. But going to this conference kind of led me to come back and get involved in my university. So I joined this committee called Women in STEM, which was just kind of a place for people to explore, people from all genders and backgrounds to explore their interests and passions in STEM. And we learned a lot about how to network and career advice and stuff like that. We went on a couple trips where we could learn about careers that were available to us in Toronto and stuff. Recently, I saw that they're doing some equity, diversity, and inclusivity training. But also, it was interesting. We learned a couple of things about interviewing, and which was something that I didn't know about until recently. Stuff like, your interviewer is not allowed to ask you your age or if you're planning to become pregnant soon, which I think is, important to take note, because that wouldn't be fair if they excluded you for those reasons. But then after coming to the University of Montreal, I joined this association for students at my research institution that I'm on with SWATI right now. And I thought this was a really great position or a great opportunity. And I think my previous experiences in these conferences led me to try a leadership position and get a bit out of my comfort zone and, meet like-minded people. I wanted to try my hand at having a bit more responsibility. And I think this was a great opportunity because I could learn a bit more about other things happening at the Institute, such as this gender and equity committee that we have, and agree steering committees. So I think this was a really exciting opportunity for me to try, and I encourage everyone to try their hand in a similar position if they can. But I think finally I'd just like to end it on kind of some tips that I have for moving forward and kind of encouraging like a welcoming environment for women in STEM. But I think it's important to stay educated as you are by attending a conference such as this, and staying involved by joining organizations that your school might have, maybe such as like an equity organization or this United Nations conference that I talked about. I think it's important to find like-minded people who are passionate about the same things as you are, to kind of, strengthen your voice, but also just some advice that I've heard from a lot of professors who have told me that it is equally as important to interview your employer and your potential coworkers as it is for them to interview you if you have the chance. So in the context of research specifically, I've had many professors tell me, don't be afraid to go over and interview your potential future lab members and ask if they're happy. And I've had professors tell me that they've rejected some positions at Stanford and Harvard because the lab members that they were potentially working with weren't friendly or weren't inclusive. So I think that's interesting to think about as well. I think it's important to consult resources at your school when you need, and have tough conversations if you're feeling like not okay or not included. But regardless, I think it's just important to attend things like this and share the knowledge that we have. But to say thank you for listening as well. And I'd be happy to take any more questions if we have any.
Well, thank you so much, Hilary. That was actually, that was quite inspiring. Even for me, that was really great. Does anybody have any questions that they would like to ask? I don't have my hand over anybody's name like I did with Chris before, so I'm pretty sure I'm not missing any. Oh, there. Although
I'd be happy to talk more about science too.
There are some hands clapping, but those don't shoot up to the top. Let's see. Just double checking. I don't see any questions. Any further questions? So where do you think you're going from here?
I think I like to start, like for me personally, career-wise, start working for the company that I mentioned that my boss is running, the Excel Thera. So I think that'd be a really nice transition, and it's definitely a field that I want to pursue. So I think that could be a great fit. And yeah, I could stop sharing too.
Oh, look at that. There's everybody. Well, if there are no further questions, takes a while sometimes to type a little bit, so
All right.
always pause a bit. Okay, well, thank you so much, Hilary. That was terrific. And you know what I'm going to do right now? I'm going to do this.